Hello everyone, I'm Shilu Zhang from University of Wisconsin-Madison. Today I will talk about defining cell type varying networks of chromatin marks and transcription factors underlying cell fate transition dynamics. A regulatory network is a set of macromolecules consisting of proteins such as transcription factors, signaling proteins, and RNAs that interact to control the level of expression of various genes in a given genome. Chromatin marks also play important roles in regulating the gene expression. At a high level, we are interested in understanding regulatory network dynamics on cell lineages. Such lineages can, for example, specify how cells transition from a pluripotent state, such as embryonic stem cell, into a more differentiated cell like macrophages. Several papers have measured cell type-specific data to study the dynamic regulatory networks during reprogramming or differentiation. The challenge, however, is that regulation of gene expression is complex, especially in higher eukaryotes. Signaling proteins, transcription factors, interactions of enhancers with promoters in a three-dimensional space, and chromatin states have important roles in controlling the downstream gene expression. These can all be potential regulators for the target genes. Regulatory genomic datasets are becoming more available, for example, ChIP-seq for measuring histone modifications and transcription factor binding, RNA-seq or macroarray for measuring gene expression levels. But we don't have good methods to analyze these datasets, especially to see how different regulators interact with each other to impact the overall expression state. In particular, there are two key challenges in understanding the role of transcription factors and chromatin marks in cell type-specific expression. The first one is that we need methods to capture the relationship between chromatin marks, transcription factors, and expression levels in one cell type or context. Another one is that we need methods to understand how changes in chromatin state and TF binding influence changes in the downstream gene expression levels. Existing computational approaches have either modeled one cell type at a time and learned one network for each cell type separately, such as SPCNs, or have merged the data from multiple cell types into one and learned a joint network across multiple cell types containing both within-cell network and cross-cell network, like ChromaNet. The key limitations of these approaches are that they cannot capture dynamic changes among chromatin marks and TFs across multiple cell types, and most of these approaches focus on chromatin marks only, but not TFs. To overcome these limitations, we present cell type varying networks, a probabilistic graphical model-based approach that uses multitask learning to capture the relationship between chromatin marks, TFs, and expression levels in each cell type, and also model the dynamics of these interactions on a lineage. This enables us to understand how changes in chromatin state and TF binding influence changes in expression levels. CVN takes as input a cell lineage tree and cell type specific data of chromatin marks, chromatin accessibility, expression, and TF binding. Here is an example of the data matrix for CVN. The rows are the different features and the columns are the different genes. CVN learns connections between the different features and how they predict expression in a cell type specific manner using a greedy graph learning algorithm together with a structure prior to enable sharing across cell types. The structure prior follows a cell lineage tree and captures the similarity of two networks from two related cell types. and the output of CVN is a set of cell type specific networks for each cell type. To assess the performance of CVN, we generated data from 4 Gaussian graphical models learned independently from data in 4 cell lines. So here is the cell lineage tree. We first use a simple probabilistic process of network structure evolution to simulate the network structure based on 3 main parameters: the probability of having an edge in the starting cell, conditional probability of gaining an edge that does not exist in the predecessor cell, the probability of maintaining an edge that exists in the predecessor cell. We simulate networks of 352 regulators for these 4 cell lines. Here are the simulated networks. Next, we generate the adjacency matrix of these networks as the precision matrix and apply graphical assert to estimate the covariance matrix. Then, we sample data from 4 multivariate Gaussian distribution using input mean and estimated covariance matrix from step 2. Here are the simulated data for these 4 cell lines. The rows are the regions and the columns are the features. We compared CVN with an existing approach, ChromaNet. ChromaNet is a statistical method to infer a chromatin network by inferring conditional dependency relationships among a large number of chip-seq datasets. ChromaNet learns undirected networks, while CVN networks are directed. So, we need to transform CVN networks to undirected networks, first by taking the max edge weight among the 2 directions, and then run by the edge weight, and select the same top edges for both of them. We generated 50 random subsamples of the simulation data, learned a network from each subsample, and computed a confidence for each edge representing the fraction of inferred networks in which the edge was present. We estimated precision and recall at different confidence thresholds for inferred edges compared to the 2 networks and plotted the precision recall curve. The PR curves for these 4 cell lines are shown on the left. Blue is ChromaNet and red is CVN. We see that CVN curve is above the ChromaNet curve for all 4 cell lines. The area under this PR curve gives an overall assessment of the quality of the inferred networks. We computed the AUPR and plotted on the red. CVN networks are able to recover the interactions in the simulated data with an AUPR ranging from 0.6 to 0.68 and better than ChromaNet. This suggests that CVN is able to predict the true interactions accurately. Next, we want to see how CVN performs in real data. We downloaded chip-seq of 9 different histone-modifications, attack-seq and RNA-seq data at 4 different stages of mouse embryonic fibroblast reprogramming. These 4 stages are MAV as the start state, MAV induced for OSKM expression for 48 hours, denoted as MAV48, pre-iPSC, and embryonic stem cells as the end state. Signals of chromatin marks and chromatin accessibility were aggregated around the transcription start site of genes. We quantified the accessibility of 342 different TF motifs using the attack-seq data. We applied CVN using a total of 353 features on these 4 cell lines and learned 4 networks for each cell line. We want to look at the differential interactions across these 4 stages during the reprogramming process. An edge presence and absence pattern can be represented as a vector of binary variables. And here on the left, each row represents a different pattern. One means present, and 0 means absent. So for example, this first H is present in ESC but absent in MEF48 and pre-APSC. This third H is MEF and MEF48 specific. To gain insight into these different patterns of subnetwork across cell lines, we conducted clustering analysis. Like in our simulation, we first generated 50 random subsamples of the chronis data, learned a CVN network from each subsample, and computed a confidence for each H, representing the fraction of inferred networks in which the H was present. Then we merged this confidence score across 4 cell lines and did k-means clustering on this H by cell matrix to find different patterns across cell lines. We choose 12 clusters, which several of these subnetworks exhibit different patterns of conservation. So for example, cluster 1 is conserved across all 4 cell lines, and cluster 2 is pre-APSC and ESC specific, and cluster 11 is ESC specific. Next, let's first dig into the Celltype specific connections in cluster 2, which is pre-APSC and ESC specific. We visualized the subnetworks in Cytoscape and found a large number of the Celltype specific interactions in pre-APSC and ESC but were absent in MEF and MEF48. The width of the H is proportional to its confidence score. Here is a zoom in of the pre-APSC and ESC subnetworks. The more red, the more they are pre-APSC and ESC specific connections. In both networks, we can see that chromatin marks like H3K9 acetylation, H3K79Me2 are interacting with different transcription factors and affecting gene expression. In particular, we found some important transcription factors such as SOX2, which is a pluripotency marker crucial for maintenance of pluripotency and controls the expression of a number of genes involved in embryonic development. And ZFX, a zinc finger protein, is shown to control the self-renewal of embryonic and adult stem cells. In the network, we can see that SOX2 is interacting with active histone mark H3K27 acetylation, and ZFX is interacting with active histone mark H3K9 acetylation, which is very interesting. Next, we further dig into the ESC-specific connections in cluster 11. Here we can see that there are more red edges in ESC but were absent in MEF, MEF48, and pre-APSC. Here is a zoom in of the ESC-specific network. The more red, the more there are ESC-specific connections. Besides key histone marks like H3K27 acetylation, H3K4Me3, we found important transcription factors such as KLF4, a pluripotency marker which regulates the expression of key genes during embryonic development and plays an important role in maintaining embryonic stem cells. And ESRB, another core pluripotency factor, also plays important roles in maintenance of self-renewal and pluripotency of embryonic stem cells. And knockdown of ESRB reduces ESC self-renewal efficiency and promotes differentiation. In summary, CVN is a powerful framework to infer cell-type specific interactions between chromatin marks, transcription factors, and expression, and changes of regulatory networks that explain cell-type specific expression levels. CVN is able to predict the true interactions accurately, and CVN can capture both the cell type specific interactions and conserved interactions across cell lineage. I would like to thank my advisor Susimita and everyone from the Royal Lab and our funding resource. Thank you. I'm happy to take any questions.